So I think I might be online already. Yes, it seems to be working. Just give me a second until I got everything sorted out. Okay. Let me see. I think I can see myself now. Let me see. Yes, I can hear myself as well. Okay, I think we're ready to go again. Hi, everyone. Hello and welcome. Yeah, face mites. That's our topic today again. Not the first time that I talk about that topic, but maybe some of you are not yet familiar on how to catch and observe face mites. So I decided, why not give it another try? As a matter of fact, here on this very microscope slide, I've got 3 face mites. 2 of them are alive. One, unfortunately, did not quite make it. And before I say hello all around the world, I'm just going to show you, this is the first face mite. Then over here, down here, this here is a dead one. Okay, it's pretty transparent already. And then I think a third one is over there somewhere. I don't know exactly where. Yeah, I have to find it. But in any case, there are three of them around here. Let me go back here. I don't know. Yeah, in any case, I think it's a little bit further up. Here, that is, yeah, maybe there's a third one. Let me find it. Here it is. Here's the third one. This one is also alive. Okay. Yeah. Basically, I collected them one by one from my eyelashes. I'll show you how to do that today, and then I all transferred them on one slide. And here they are. And they have now been alive for a couple of hours because in preparation of this live stream, I've been, been preparing them. And where is the other one over here? Here it is, right? Okay, yeah. So that's basically what I'd like to do today. I'm going to basically just have a quick look in the chat here. And I'm going to spend the first couple of minutes. I like to always say a little bit hello all around the world. It takes a couple of minutes for people to join into the live stream. So from England, hello, okay. From Northern Germany, sound works, thank you. From Germany again, Sri Lanka. From London, the United States, France, the United States, okay. From Paris. Okay, here we are. The question you might answer is, can we not make it a little bit larger, this one over here, right? It's, I'm using over here, as you can see, it's my 20 times objective, and I said not yet, because there is no cover glass on here yet. I'm going to be adding one later, because the face mite is in a drop of oil, and because I needed access to it because I was using a pipette, and therefore there's no cover glass on top, and therefore I cannot go up with the magnification further, okay, for safety reasons. So what magnification is that? I'm using now my 20 times objective. I'm going to now, just as a reference, later on I'm going to show you all of the, how do you say, from the very beginning, okay? But just that you understand a little bit how big it is. This is now the 10 times objective. I have to go down a little bit, And what you have over here, this is a hair. Yeah, that's one of my eyelashes. Yeah, just so that you see a little bit the size difference. And the reason why this eyelash is on here is because of something quite interesting. I'm going to use the arrow right now, because this face mite over here, which is now a little bit blurry because it's out of focus, used to be in here. Okay, and then it broke free, and then it's now basically over here, right? But you see that the diameter of this face mite is less than the diameter of one of my eyelashes. Yeah, and this is basically the place where the eyelash is inside the skin, so it's like the hair follicle, and the face mite is living right here because it's a very fatty and oily place, and it feeds on fats and oils, right? So that's kind of, and let's go down further. So that is now the lowest magnification is the 4 times objective. I have to swing out the condenser here so that you see this, right? So that's, you see over here, that's again the eyelash. Yeah, that's the face mite. And this one down here, that's the dead face mite from before. Let's move it up a little bit. Yeah, you see, it's interesting. The dead one is more transparent. It used to be darker. It was alive before. For whatever reason, it died, and then it became somewhat more transparent over the course of an hour or so, right? And the third face mite that I have got is over here. Yeah, that's the one that I had some problems finding, right? Yeah, so this one is also alive. So we can go up again with the magnification. Here on the side, what you see, that's the edge of the oil droplet. So I've got some vegetable oil, some cooking oil in here. And I'll show you basically how I'm doing that, okay? So let's go up a little bit again with the magnification, right? And so that's, yeah. So a little bit... and 20 times and higher I'm not going to go yet, okay? Later on I'm going to add a cover glass and I'm going to go up all the way, right? Yeah, so I'm just reading some of the comments here. By the way, if you're kind of new to this format, please feel free to comment. Please also say at Microbehunter or at Oliver if you want to directly talk to me, because sometimes there's so many comments that I have a problem actually reading all of them, okay? So I'm going to now basically, as we observe those, another organizational, a few organizational aspects. I've got a little cold, so maybe you can hear it on my voice that I've got I have a little cold right now. I'm feeling better, but just in case you're wondering if my voice is not quite as clear as it normally is. And another organizational thing is, honestly, I can show this to you in 10 minutes and then we're finished with a live stream, right? Okay, no, so please do feel free to ask questions to keep it going a little bit. And in case we really run out of things to talk about, I do have also some other specimens which are non-face mite related as well to show you. But I do not want to hurry, okay, about this. Yeah, so that is basically what I would like to show you. And the question is now how do you actually get them and what are they even and so on. So those face mites, if you are young, a child or an adolescent, chances are that you do not have as many of those. The older you are, apparently pretty much everyone's got those, right? And what they're basically they're living in the hair follicles. The hair follicles are basically the cells that make the hair, right? In this case, the eyelashes, and this is where they live, right? They basically lay eggs. They reproduce. They lay eggs, and they just live there and they don't do anything, right? I don't even know if you can even call them parasites because they don't actually do any harm or anything, right? Yeah. I know that in some cases, if there are too many of them, they can actually cause some irritations called rosacea. But otherwise it's kind of, they don't do anything. More older people have them because generally older people produce a little bit more fat and oil, and they like to feed on fat and oil. Yeah, there are different types of species as well. Some are larger, some are shorter, and basically, yeah, they're part of our normal ecosystem. Yeah, on the body. Yeah, the interesting thing is that they do not have a fully developed digestive system. So basically, they have a mouth. They eat the oil, but essentially the waste are kind of stored inside of the body. Yeah, so they're not, in that sense, they're not pooping, if you want to use that type of expression, right? In that sense, they're quite interesting as well. I did a little bit of reading. There are two species, yes, yeah. One is in the hair follicle, the other is deeper in the sebaceous glands where the oil is made. Yeah, there are different types of species. Honestly, two species have been characterized. There are, to my knowledge, there are even more species, of course, different but not necessarily on humans, but also other animals also have them, and they might be different species living on other animals. Yeah. Interesting. Recent research has shown that they do have an anus, though it is still widely unknown. Interesting. Okay. So, yeah, biology progresses as well. So how do you actually, what materials do you need and how do you get them, right? So I'm going to run you through the materials quickly. So I'm going to show the desk view again here. Okay. There are basically many more species. Dogs have their own, cats and so on. Yeah. discovered back in the, I don't know, 19th century or so. A doctor put, I think there was some kind of a pimple in a patient, and he put some of the oily pus under the microscope, and he saw quite a few of those. And by counting the legs, they quickly identified that actually this must be some kind of a mite, because mites have 8 legs. Yeah. Do they eat any kind of oil? Well, let's put it this way: at least they eat the kind of oil that we humans produce, right? And I don't know if any. The skin is the natural place, so I don't think it's if they have been. If you're able to reproduce them outside of the body and give them some other oils, I don't know if this actually has been done before. Yeah. So what I would like to show you is you need some sticky tape. Okay, let me focus this a little bit here. Okay, that's kind of clear. You need some sticky tape. I'm going to show you how you can extract them from your eyelashes. I like to use, you need those tweezers, and you see those tweezers here are pretty flat. Okay, oh, they are found in pimple pus. Ever Smaller is asking. I will have to try this method. I did find the original paper. I think it was written in German. It was a couple of years ago, a scan. And this doctor actually put some pimple pus of a patient under the microscope, right? There was some kind of an infection that the person had. And he put it under the microscope. He made some drawings, and there were actually several of those in there, right? So I'm not saying that all pimple pus, all of them have it, but at least that's how this doctor kind of discovered it. And he made some drawings. Photography through the microscope didn't exist yet at that time. Then he basically showed it to some zoologists, entomologists, and whatever, and they said it must be a mite. Because of the 8 legs, so that's kind of how they identified. Yeah, so that is those tweezers here are used usually for if you're collecting stamps, for example. People use those. You can also use those tweezers for placing a cover slip. However, I will be using those to pull on my eyelashes. That's why they're so flat. You need, of course, microscope slides. Okay, and here, this is regular. You don't see it because it's just a couple of drops. This is just regular vegetable oil. Don't use immersion oil for that, because immersion oil does contain, I think, some substances that might not like. But this is just regular vegetable oil, and it works. And we're gonna use this vegetable oil to get the face mite off the tape again. Okay. So what I'm gonna do is I'm gonna show you what I have done, and because most likely you need a mirror to do it properly, and most likely I'm not gonna be able to find now face mites, because it does take a little bit of trial and error. For this reason, I've prepared 2 slides here which actually do contain a face mite each, because I checked. Okay, so in case I'm not able to extract any face mites now or find any now, we're going to be using those slides here. Okay, because how likely are you able to find face mites? Well, today I was able to find 1, 4, 5, 6 face mites, and I made approximately 10 slides. So about 50% success rate. Right? And considering the fact that it's quite fast and easy to actually extract them, you should be able to find one in a couple of minutes. If you're lucky. Yeah. So what do you have to do? I prepare a little bit of tape here, a longer piece of tape. Where am I going to put it? I'm just going to put it here now, just for right now on the side here. Okay. To keep it for later, and then I need 2 smaller pieces of tape as well. Okay. 1 and 2. And now comes the trick, and I will tell you what I've done before. And if you check my channel, then you're gonna find some older videos where I've used the old method, which I do not recommend because it's not as efficient. So what I've done before is that I took some tape and I taped it on my forehead, and I left it there for half an hour, hoping that face mites are going to come out, especially if you keep it dark at night, then they're going to come out, and then from your skin, and then you kind of peel it off, and then you kind of hope that now I can't even get it off anymore. and then you kind of hope that there are face mites on here, right? This method does work. I've tried it before, but it's very inefficient. So that means you have to make many trials just to find maybe a single face mite, right? If you're lucky, yeah. And I think the reason is because there are not so many hair follicles here, right? And maybe that's also not the place where you can find a lot of those, but you find quite a few of them in your eyelashes, right? So I'll be using the eyelashes. I mean, I can still just for the sake of it, I still can yeah, put it on here, yeah, to keep it a little bit, yeah. So how does this work? So what you do now is now I need to move a little closer. You take those tweezers here, right, those flat ones, and you carefully try to grab some of your eyelashes. You pull and you carefully rotate. Okay, and you keep on doing this maybe for, I don't know, a couple of seconds or a minute. Yeah, don't rip them out, but you carefully rotate. And this method is not a method that I invented, but this is what some doctors are using to actually get them out. They don't use those tweezers, but they have different types of tweezers. So they're kind of wrapping around yeah, the eyelashes individually around those tweezers. And they're doing that, and this squeezes out the face mite. I'm going to show you a little bit how this works. If this is kind of the how do you call this, the hair follicle, and in here you've got the face mite, right? And by rotating the hair like this, you're squeezing the face mite out. Yeah, it's kind of the pressure kind of squeezes it out. Okay, and now what you do is the next thing. Once you've done this for a couple of minutes or a couple of seconds, you do the following. You take the tape here and sticky side on the outside, and you wrap it around a pencil, or I'm just using the back of my tweezers here, yeah, like this. And then you use this edge here, sticky side on the outside, and you have to now look here what I'm doing right now, and you kind of touch and you try to collect, and don't do it too lightly. Make sure that there's good contact. Okay, make sure that there's good contact. And yeah. And this is a little bit of a question of luck as well. Okay, I'm just going to leave it at as it is now, okay? And what you have now is you basically have got a line here where hopefully you'll have collected a face mite, right? So that's we have to find this out. And what you do now is you take a microscope slide, okay, and sticky side upwards. Sticky side upwards. You're now using those two smaller pieces that you prepared, and you kind of stretch the tape across the slide. And over here, somewhere in the center, this is difficult to see now. This is where you've collected or should have collected now whatever you've had on your eyelashes. Okay. So what I'm going to do now is I'm just going to put this now under the microscope, and I'm just going to be searching this central part here, right? Yeah, let's have a look. Let's go back to the scope view. Let's see. It takes some time for it to switch. Oh, okay, here we go. Yeah, that's the face mite from before. Okay, and again, if I'm not able to find any face mites on here, then that's fine, because I've prepared two other slides where they are. Okay, so let me put this over here somewhere into safety. Where am I going to put it? To safety over here. So, and now I'll put in the one, yeah. Okay, I'm going to remove the DIC. Let's go down with the... Okay. I have to find now the place. And every time when I see something long like this, you might want to have a closer look, okay? I don't know if this is one or not. I have to go up a little bit with the magnification. It doesn't... Oh, it could be one. I don't know, but it does look a little bit. It does look a little bit distorted if it is one. But actually, it looks a little bit broken. But it could be already one. But let's do the following. Let's have a look here, and you want to find the place. Let me see. I don't know where I. You want to find the place where you see a lot of skin cells. You see all of those little round things here. These are all skin cells. And that is basically, those skin cells are actually the how do you say, indicates that this is the place where I've actually had contact with my eyelashes. Yeah. And every time when you see something long, then you might want to have a closer look at it. Yeah. So there is something longer over here. So I'll be zooming into that one to have a look. Okay. Yeah, and no, that's nothing, right? Yeah. So let's have a look here. Yeah, so usually what I do is I kind of scan back and forth. And then every time when the density of the skin cells goes up, then I know that I'm basically in the area where I actually collected. And then over here, the density of the skin cells goes down again. Yeah, so I'm kind of, but I wonder if this is what I saw before. Let's go down with the magnification a little bit, because this allows me to scan everything a little bit faster. So we're going to be checking the place where there are lots of skin cells here. Okay. So, yeah, over here as well. Lots of skin cells are here as well. So, and you see that it's a little bit difficult to actually distinguish, see the different structures here. So I'm going to show you now a little trick how you can actually make the face mites, if they're present at all, more easily visible. Okay, and that is you directly add a little bit of oil. Okay, you add a little bit of oil directly, and this will make the skin cells go transparent, but the face mites, if they're present, will be visible much better. Okay, so I'm going to show you this. So what I'm going to do now is I'm going to, I don't even remember where the one suspected face mite, oh, this one is also a long one here, but it doesn't look like one. Okay, I think it was here somewhere at the top. Okay, if we don't find one, don't worry. I've got 2 more slides that actually do contain face mites, because I prepared them and I checked that they are actually on here. Yeah, you always want to remember a little bit where you found the things from before. I wasn't quite sure whether this one, what we saw before, actually was one or not. Yeah. But essentially, if you don't find one, you just kind of repeat the whole thing again, right, until you're more successful. But let's add a little bit of oil. Let's see if this one, I don't know, no, this one was the one, yeah, before. Yep. So that's what I mean when I say you do need a little bit of patience. Yeah. So let's do the following. Let's add a little bit of oil. Okay. So I'm just gonna take my pipette. And I'm going to go down with the magnification a little bit so that it's actually, I have a larger, how do you say, place to work with. Okay, you see how messy everything looks like. Okay, let's remove the arrow here. And let's add a little bit of oil. Up. Okay, you can immediately see that where the oil kind of touched it, it becomes more transparent. You know what I'm going to do is I'm going to remove this and I'm going to show you directly on my desk what I'm doing. The desk view here. All you want to do is you want to take a little bit more oil maybe. Ah, the tip fell off. Okay, all you want to do is you want to add a little bit of oil and carefully spread it. because of the refractive index of the oil, the skin will become more transparent. The oil will also dissolve the glue a little bit. And yes, let's go back to the scope view. Still haven't figured out, because for whatever reason it takes quite a long time. You see, everything is much more transparent where the oil is, so the skin cells are not quite as visible anymore. Okay, and then if there are any face mites present, and I don't know if this one here, for example, is one or not. If there are any face mites present, then they will be much better visible. Okay, but I have not, have not been able to find anything. And then in a minute or so, I'm going to read again some of the comments, because I just have seen that somebody commented. Dave commented with UV activated resin to make a leaf peel. That's to make the surface of leaves visible. That's interesting, yeah. So every now and then we have to refocus. Yeah. So as expected, over here, I just wanted to show you no face mites here. So what I'm going to do now is I'm going to just move over to the other one, which actually do contain some face mites. Okay. But I'll do that after having quickly read through your comments. Okay. So very quickly, so far there are no questions for me. Okay, a comment of allergy or sensitivity to the tape adhesive. So, because some people, when they get the glue on the skin, might have an allergic reaction. Okay, Dave says about experimenting with leaf peel using UV activated resin. I did not try that yet. Okay, that's interesting. There is also called this nail polish that you can use, which is UV activated. Yeah. So let's do the following. I'm going to. This was the one that where I was not able to find one. I'm going to put this away, and over here, I'm just going to show you. I've got another one, and let's try to find the. Let's try to find it here, and then let's try to also add some oil. Okay. You see that's very dense with the many cells. But I think I almost forgot. Okay, yeah. So have a close look. Are you able to see it already? So that's now just as a reference. It's now using my 4 times low power objective. Okay, this one over here, that's a face mite. Yeah. So let's easily, it can be overlooked quite easily, but you see, even at low power you can actually see that there is something there. You have to zoom in. We go up with the magnification, and now we can already see that there is already the basic anatomy. We go up now with 20 times, and here it is. Okay, you can see the 8 legs. You can see the 8 legs. It's kind of folded here. I don't know if it's still alive or not, but my hope is now to get it off and get it so that I can transfer it to a different slide, right? And that's what I would like to show you next. Okay, how am I able to get it off? So first of all, those face mites, they're hydrophobic, so they like oil. They're covered in oil, so I'm not able to get it off by adding water, so I have to add oil. And that's basically what I'll be doing now. I hope this is again takes a lot of patience and a steady hand, and maybe I'm going to be successful or not, I don't know, but at least you get the idea. So what I'm going to do now is something like this. I'm going to, and I'm now going to take a little bit of oil, and I'm now going to add a small drop of oil on top here. So I take, you can actually see what I'm doing at the, up here. So what I'm doing is I'm taking the tip, I'm resting it on the tape to give it some stability, and I'm kind of moving forward until I hope to be able to, here, see, that's the tip. and I press. And up, now it just got, yeah, covered it. Yeah, okay. So you see, because of the refractive index of the oil, I'm not able. So now I have to find it somewhere. Here it is. Yeah, do you actually see it over here? Yeah, but it also looks very transparent, just like the dead one before. So it might be dead. Okay, because for whatever reason, the dead ones are more transparent. So I'm going to see if I can actually go up the magnification. Yes. So, yep. Okay. Yeah. So this one actually is a little bit unfortunate, but this one is not alive anymore. It looks pretty transparent. But maybe I'm still able to remove it somehow, simply to illustrate how this works. And what I have to do now is I have to use the pipette again. And with the pipette, let me flip over the condenser again. It's difficult to see now. I'll be adding a little bit more oil. And then I would like to suck off. And then you have to hold. It's still there. Okay, it's still there. It doesn't want to get off for whatever reason. You can see it over here. That's the one. Okay. Maybe because it's already so dead and flat, and for this reason that it doesn't want to get off. Let me give it another try. See, I'm going to be looking through the microscope myself because it's simply easier. Okay, I can't get it off, but it's dead anyway, unfortunately. Okay, that's something that I did not expect. I thought it's going to be alive. But when it's alive and you add oil, then sometimes, at least this is what happened before, it was able, because of its movement, able to break free. And then it was kind of floating around, and then you could simply suck it off and transfer it to a new slide. But let's try it now with the other one, and I hope that the other one is alive as well. Okay, because otherwise we're just gonna keep on observing the slide with the two live ones. Okay, let's move the arrow again away here. So. So again, you see a lot, a high density of cells here. Yeah, and what we're going to be doing is we're going to have a look again at those dark, larger structures. And this one here also looks like, at least I think it is one. I don't know. Yeah, almost certainly. I do hope that this one at least is alive. Otherwise, yeah, yeah. And we're going to be doing the same thing. And again, if it turns very transparent, then we know that it's probably dead. Let's do the following. It's, yeah. So. Because sometimes when you add oil, sometimes it looks also transparent. Oh, that's a pity. Okay. It could be, and I guess it's a pity. Yeah, it could be that maybe if you actually do that right away after you've taken it off, maybe it's able to survive then. But this one, that's a pity. Okay, yeah. So kind of let's just assume that they were alive. Then I would pick them off, and then when they're picked off, then I would transfer them to a new slide, like the one that I have over here. And so that's my eyelash again. And I have to find it again to focus. Yeah. And somewhere, yeah. Is this the one from the dead one from before? Okay. Let me have a look at the low power. Yeah, here is one. Let's have a look if this one is still alive. Oh, it's still moving. It's still moving. Okay, this one is still fine. So what I would like to do in this case I'd like to I'd like to go up with the magnification yet further, so it means I have to add a cover glass. And there's a question, I wonder if face mites have a skin like tardigrades. Tardigrades, I think, have a skin made of chitin. And I think face mites must have as well. Okay? So that's kind of the thing. And what I would like to do now is we're going to have the whole thing now look under the high power, and for this reason I need to add a cover glass. Okay, so let's take it off, and desk view. So it was not quite successful with the dead ones, and let's see if I have a cover glass. Let's see if there's enough, not too much oil, not too little oil. And of course, there's always a little bit the danger when I add a cover glass that of course everything spreads. And then there is of course the possibility that you won't find it there anymore where it was before. Yeah. So let's see. Is there only one of them? Okay, yeah, there's only one. So I'm just gonna drop it on here. So everything's gonna spread a little bit. And I hope that it's not going to completely crush them. And now I have the challenge of finding them again. It takes some time for it to... Okay. Yeah, here is one. This one is still pretty much there where it used to be. So let's go up now with the magnification. It's a little bit now still from the side. So 20 times. Okay. And 40 times. DIC. Let's see if this adds anything here. Not a lot actually. But I would like to have a look now using phase contrast, because maybe this actually does improve it a little bit. Yoop! Wow. Okay. This is actually quite contrasty. Actually, phase contrast works quite well, but the depth of field is a little bit off. So you see that because I'm not able to close the condenser, by closing the condenser I could actually increase the depth of field, but in phase contrast I have to keep it open. So that means part of it is always a little bit blurry. But okay, what I'm gonna do now, I think I might actually look for, that would be dark field now at low power. So, but let's see if I'm able to find the other one. Where was the other one? See, everything kind of... I wonder if I'm able to find the other one. Because when I added the cover glass, of course, everything shifted a little bit. Otherwise we just, here it is. Here it is. Maybe this one is better. This is a top view. A little bit better, I don't know. Yeah, so it's also a little bit, yeah, blurry because of the low depth of field. Let's stay with this one here. DIC. Let's close everything here, the condenser. Yeah. This gives me a slightly... So we can go up yet one magnification step further, 60 times. And that's not a largest... I'll not use oil immersion, but that is not the largest magnification here. Yeah. Yeah, so those have actually been now, those two have been alive now for a couple of hours in the oil. Yeah. Yeah, so, and in the past I've seen all sorts of them already, the larger ones, smaller ones, yeah. Yeah. So let's, but yeah, DIC, but it's interesting if I change the, let me change the colors a little bit maybe. It doesn't really, it's just playing around now. Yeah. I mean, I don't know. I don't know what you think. I consider this fascinating. I mean, critters like this growing in our body. The world is so complex, you know. There's a world inside the world. Yeah. How can the face mite breathe if it's literally drowning in oil? Well, yeah, it takes up oxygen. Of course, it needs oxygen. It takes oxygen over the skin, but because it's so small, it has a very large surface area to volume ratio. And do not forget one thing: oil is hydrophobic, right? Yeah, and so is oxygen. Oxygen actually dissolves. Yeah, it dissolves in oil. Oxygen does not quite dissolve as well in water because water is hydrophilic. Yeah, so in other words, oxygen is non-polar and oil is also non-polar. So I can imagine that the oxygen actually has no problem going through the oil. By the way, if you're a little bit buff biology, that's the reason why, for example, we need red blood cells with hemoglobin to actually carry oxygen, because the oxygen does not dissolve very well in water. Yeah. So in that sense, I think there are multiple factors that kind of play a role here. The large surface area to volume ratio of those small animals, and also the fact that actually the oxygen is able to diffuse through the oil. Yeah. Yeah. So. Yeah. So this is not time lapse. Okay. This is actually now real. Real speed. Yeah. The movement does not appear quite as smooth as you just see, and I think this has to do something with the shutter speed of the camera. Yeah? Have you found any nymphs with longer tails? I'm going to show you now something, a video, okay, that I made just in preparation here, just to show you. Okay, video 1. So this is a video, and look at this one over here. Yeah. I kind of just demonstrated this to you. This is a video, right? Just in case it didn't work, right? And you can actually see, where is the arrow? Yeah. This one over here, this tiny one I was sucking up. Yeah, and this one was the one from before. And I added the other one as well. Yeah, and that's basically, these are the ones that I found. So basically I put both of them together, and I'm going to pause the video now. Where is the video? Where's the pause? Because I want to talk a little bit about this. This is actually one of them which then later died. So that's the one that you've seen, yeah, became transparent. And this one over here, you're going to laugh. After 1 hour, I couldn't find it anymore. Seriously, I actually lost it. So what I think now is maybe it also died and got transparent, and therefore I was not able to rediscover it. But yeah, but you see that there are different shapes of these here, right? So this is yeah. Let's continue with the video here, because I just want to show you something else. where do I start the? Oh, here. Let's go on. Okay. Yeah. So that's basically this video is just a few hours old, right? Yeah. Here, larger. Yeah. Yeah, and that's basically what we've just seen as well. But this one is a video, right? But there is a second one that I want to show you. Okay, so far nothing unusual. This is just nice because it's a top view and you can actually see the 8 legs. You can see it's a little bit segmented, those tiny lines here. Okay. So, and video 2. And this is cool. This is the hair follicle. Do you see this here? I saw this tail here. It says, wow, there is still one of them here connected to the hair follicle, right? The video is now looping. That's why it's kind of jumping around, right? But then later on, this one was the one actually which it started to crawl out and was separate. Yeah. So this is kind of, at the beginning it was still here in the hair follicle. And then later on it was able to go out and then it was next to this here. So let's have a look here at the scope view again. Let's go down a little bit with the magnification again. Okay, let's go again down with the magnification. I need to find it. Where is it? Where was the... I forgot where... Where was the hair again? Just a second, I need to quickly check. Over here it is. Yes, here we go. That's the one I wanted to show you, right? That's the same hair follicle from the video, but now you see that the face mite is gone, right? And where can you find it? Down here. Okay, yeah, so it was able to crawl out. Yeah. So just a little bit of a before and after, but I'm not going to read read a little bit again what your comments are. I'm kind of happy that they're still alive, so that we're able to see them. Have you found any nymphs? So I don't know if this one with a long thin tail was actually a nymph or not. Yeah. Honestly, unfortunately, I don't know. Yeah. Don't forget to be asking questions. Yeah, yes, please, because otherwise, I don't know how to fill up the live stream. Hold up, one of these face mites has a very long tail. Yeah, that's the one that I just showed you, which then I was not able to find anymore, right? Yeah. How long does a face mite live? I don't know. I read it somewhere, and because what they do is they keep on reproducing. But that's something that can be, I think, easily researched. They keep on reproducing, they crawl out at night, they mate, they lay eggs, and then you have a next generation forming. Yeah? Lifespan of around 14 days. Yeah. And then it's kind of, what happens to them when they die? I mean, I guess they're kind of reabsorbed again back into our body. Yeah. So that's the, so I'm quickly reading it again. Typical 14 days. Human face mites, is that a type of a human face? Well, the question Tino Coco is asking, human face mites, is that a type of face mite? There are different species on the human being. There are 2 different species, at least documented up to now. And of course, the different mite species living on other mammals. Yeah, not surprisingly. So, ever smaller, Jesse. There are more than one species there's 2 main species, then there are species on many other animals. Yeah. So it's nothing. It's not nothing really surprising that if you find a certain type of organism on a human being, on a mammal, that you find some related one also on other mammals. Yeah, so you can see that actually each body or each organism is like an ecosystem or like an ecological niche, so to say, for other organisms. Yeah? So let's go up here again a little bit with the magnification. I think I don't know. It's really. I'm fascinated by those creatures here, right? Yeah. By the way, in case you're wondering, if you're the trailer of these live streams, before the live stream starts, there's this trailer where I show some video clips and pictures with some music. There's also one of the clips is also one of these face mites, a fairly large one that I found. Yeah. So yeah. What do they eat? They eat oil. Which oil do they eat? The oil that is produced by our skin. Yeah. So that's the only food that they have. Yeah. I guess I mean, they do have a mouth, interestingly. So yeah. So they do seem to eat it in that sense. Wouldn't be surprised maybe if they also eat some other smaller particles that are in there. But mostly, what I heard is that they feed on oil. Huh? I mean, they do need other, besides oil, they also do, of course, need other nutrients as well. So maybe they're also feeding on bacteria that are present in the glands. I don't know. Yeah. Okay. Yeah. Thank you again. Ever smaller. In my reading, I would say dog ones, the Demodex canis, are the most widely researched, even more so than those in humans. Oh, interesting. Ah. Okay. Yeah. Oh, okay. I don't know if you're able to hear there's a fireworks on the outside now. Why would they have fireworks on a Saturday evening? Okay, now it stopped. It's usually, they always do that when they have a wedding on the next day. So that's to announce the wedding. That's why maybe you've heard the explosions. Yeah. So pretty nice, I think. Yeah. So I'm going to have another look at what would happen if all of the face mites died off? What would happen to my skin? Honestly, I think young children and so they don't have face mites. You're not born with them, right? They come over close contact with the mother, with other humans. That's how they're actually transferred, and then they start growing. So actually, we humans did not have face mites when we were born, and we were also fine. Right? Some people have more, some people have less, and I don't know. I can imagine that yeah, I don't know if you were to remove them. I mean, nothing's going to happen. I would say, right? They're just a normal part of our body, and yeah, and in some rare cases they can overpopulate, and then you have some irritations, maybe. Right, and what happens if they die? Well, they do die because as we just heard, they have a lifespan of approximately 14 days. Then I can imagine they're being reabsorbed. Maybe our immune system takes care of it and breaks down everything and then reabsorbs them. Yeah. Okay. Yeah, according to a quick Google search, they also eat follicular and epithelial cells alongside the sebum. The sebum is the oil. This is according to National Institution of Health. Yeah, wouldn't surprise me. If there is food present, yeah, in the form of cells, why would they not eat them, right? I would assume removing them would mean you will have an oilier face. I don't know. Don't know about that so much. If yeah, because actually whether we have a lot of oil on our face, I mean, depends generally also on our hormone. Yeah, how many hormones we have during puberty, there's more fat and oil production. Yeah, so yeah. But in any case, yeah. By the way, folks, I just want to share something with you, an idea, but I don't know how to realize this. Why not have some kind of a face mite challenge? You try to, I don't know, find face mites and post pictures online maybe, and then give it maybe the hashtag face mite challenge. I do also have Reddit, where I also cross-post the links to these live streams and the videos, but unfortunately, I think it's not possible to post pictures in beneath the videos. Yeah. So otherwise, this would be kind of nice if we could make some kind of a picture collection. Yeah. True, but if you don't have mites eating it and breaking down into waste, then you will have more oil than you would have. Honestly, I don't know if the amount of oil that they're breaking down really makes such a huge impact, considering how small they are and how much oil there is actually on our face. I mean, the interesting thing is what I also read somewhere is if you now use indeed a lot of, for example, soap to remove oil from your face, then this actually causes your skin to overproduce oil and fat. Yeah, so there seems to be also some kind of a natural response. Yeah. Shall we not wash our faces and count them? Yeah, I don't know if the thing is it would be interesting to figure out a way to actually determine the how many there are in a face. But you see, it's even if you're collecting them using the tape method, it's not always you're not going to catch all of them this way, right? So I don't know if this were indeed a very reliable way to do some kind of a biodiversity study. Yeah, yeah. Demodex follicularum lives mainly in hair follicles, and brevis lives in sebaceous glands. Ah, okay, good. Yeah. So I wonder if those here are then maybe mainly follicularum because they were in the hair follicles because I was kind of pulling on my eyelashes. I don't know. Yeah. So let me have a look. Let's go down again a little bit with the magnification. Let's check out the other one again if I'm able to find it. And here again, that's somewhere. What was the other one over there? Somewhere. Let's go down all the way with the magnification again. Yeah, it's finding them is always such. Ah, here it is, the other one. Okay. Is this one still alive? Still going as well. Still going as well. Yeah. Yeah, for those of you who maybe joined in a little bit later, yeah, let's. While I keep this running here, I'm just going to quickly demonstrate for those of you who joined in later how I was actually collecting them. Yeah, the ones that we basically already did 3 trials. The first trial I was not able to collect any, unfortunately, find one at all. And the 2 other ones where I prepared them, they already died. Okay. So what I'm going to do now is I'm just going to, we can continue watching those. I'm just going to prepare again a couple of pieces of tape. I also tried it using pulling a little bit on my eyebrows. I was also not quite successful there. Yeah. So and let's have another try. So I'm going to just grab some of the eyelashes. You can maybe also try the top ones, but the bottom ones are a little bit easier to reach. No, can I grab them or not? Here we go. You carefully rotate. If there are any present, they're going to be squeezed out this way. Maybe move over a bit. Okay, maybe that's going to be enough. I don't know. It's just, usually it's easier in front of the mirror. You take some tape, you put the tape, here it is. You put the tape, wrap it around here with the sticky side outwards. Okay, and then I'm going to touch my eyelashes, hoping to be able to collect any that might have been squeezed out. And this is trial and error and patience. Okay, so that's basically what I've done. You take a microscope slide again. I'm going to show you here quickly by switching over to the desk view. Oh, I already have a slide here. Okay. And sticky surface to the top. And you use 2 pieces of tape to secure this tape here. And here in the center, this is there is a line here where I collected the face mites, hopefully there are a few present. Okay, and okay, and we are now going to have a look again at the scope. Okay, and we're going to have a look again here. Okay, and we have to scan it again, and hopefully we're able to find something. Okay, so that's a little bit the strategy that I'm using. You again, if you just joined in, then essentially we have to find now the place where. Oh, thank you very much. The questions are here again, and then we basically scan the tape again, hoping to be able to find. Yeah, but over here I've not got anything here. Ah, yeah, here it's I think. Yeah, places where you were able to find some more cells here. Let me quickly check. Ah, yeah, here, and then if we're lucky, we're able to find some again. Okay, so that's kind of the procedure, and now basically it's then a question of trial and error until we're able to find a few more of them. Okay, so let's do the following. Let me quickly remove the slide again, and here we are back. Yeah, over here, where the one where we prepared it before, and I have to find it here again. Let me low magnification. Here we go. Okay. So, okay, yeah. So what we have to do is now the following is we don't have to do it, but I've got again a couple of questions here. But I've just realized that I've actually responded to most of them. Okay. So let me quickly have a look at the desk here. Okay. Yeah, many of them, some of the questions are already mentioned. There are 2 species. We talked about that. Do they eat any kind of oil? Yeah. I would, if you're already asking, if you already ask any kind of oil, then I would probably say no, because not every oil is edible. We humans are also not able to break down any kind of oil. I would say that, of course, the oil that is present in the skin is the one that they've are optimized to metabolize. Yeah. The reason why I added vegetable oil is because this way the glue dissolves of the tape, and they're also more easily they can be more easily removed from the tape. Because I tried it once with water, and basically I was not able to pick them up that way, because the face mite being hydrophobic, being fatty, it did not really like to stay in the water, and then I had a problem picking it up. Okay, yeah, it's the tape allergy that we talked about. Yeah, so the leaf peel, that's a little bit off topic. Okay, but it works like this, that you apply a little bit of glue on the bottom side of a leaf, and then when you peel it off, you can actually see the surface texture. Okay, you can see the surface texture of the leaf, and then you're also able to see sometimes the stomates and the shape of the cells. Yeah, so it's kind of, you're making a leaf imprint. Yeah. Have I tried looking at them under cross-polarized? No. And I have to be honest, my microscope does not allow for standard cross-polarized light. I do have a DIC microscope, that's correct, but there are prisms in there as well. And so at the standard cross-polarization I don't have. And I probably would guess that maybe you won't see a lot, because the face mites, I don't know if they're actually depolarizing the light to the extent that you're able to see anything. Yeah. But one would have to try out. Yeah. Okay. Good. Yeah, the stomates, we talked about those. Yeah, by the leaf imprints. Do they have a skin like tardigrades do? As far as I know, tardigrades have a skin made of chitin and face mites as well. Okay, so in that sense, chemically, it seems to be similar. Please, somebody research this. Huh? Yeah, how can it breathe? The reason is because the oxygen diffuses through the oil, and it also diffuses through the skin of the face mite. I don't know if the one that we've seen before it was actually a nymph. I have to tell you, I don't know. Yeah, and don't forget to ask questions. Face mites live 14 days. This has been now researched by ever smaller. Okay. Demodex canis, yeah, most of these are already addressed. Okay. Yeah. So, okay, well, there are, okay. So what I'm going to do then is the following, because I still have, let's say, around 20-25 minutes or so. Unless you want to continue watching those face mites, I would actually like to show you, make a complete change in topics, whatever you want, because I would like to share with you as well, like I said before, maybe this one over here, how I now finally managed how to make thin cross sections of very hard oak wood. Because this is something that I've been trying as well. Yeah, unless you want to to continue with the face mite topic, I would actually want to show this to you as well, because I was semi-successful with this series and I wanted to show this to you as well. So what I've got here is the following. This here is our small branches or pieces of oak wood, so very hard wood, and this is actually the wood from what they claim to be Europe's oldest oak tree, which is 1,000 years old. I visited this tree a couple of weeks ago. We had a short holiday, and I did not cut the branches off, but they were on the ground. You don't want to do any damage to the tree. And the problem is now how can I get the wood soft enough that you can actually microtome it, right? And you might already remember from a previous video this microtome here, right? But the problem is that the wood was extremely hard. So what I've done is the following. I've actually put it into glycerin. So this is 100% or 97% glycerol. And this is basically, I left it in there for 2 weeks and it got a little bit softer, but not a lot. And really to soften it, what I have done is I quickly put it into the microwave for a minute or so. And this got really hot. And then all of a sudden, the oak wood started to actually become softer. I'm just going to show you. Okay. Yeah. If you just, you can bend it a little bit. Yeah. Well, actually, it now hardened again a little bit, but after I heated it up, it was actually fairly flexible. Okay. So maybe I'm going to heat it up. Maybe I should heat it up again. It doesn't matter. I was able to make those cross sections. So, and this actually worked surprisingly well. Before that, it would have been impossible to actually cut through the wood. But now it was possible to cut through it. And I want to show you what I've done here. So this is my homemade microtome knife. Okay. It's 3D printed, as you can see. 2 screws in here. I put them a little bit deeper so that they do not scratch, right? And this is a regular razor blade. However, there is another thing that I want to show you what I've got here. Where is this? Look what something else that I bought that I'm trying to. This is also a razor blade, extremely sharp, but it has a disposable blade. And the advantage of this is that it also is very rigid, so it's not flexible, right? And I also want to show you one of those replaceable blades, how it looks like. Okay, those blades, they look like this. I mean, there are also different standards and systems here, but that's actually one of those blades that can actually go in here. right? This one here is also a bit flexible, right? But if you put it into the knife like this, it's fairly rigid, right? So, and you can use this knife, or I have been using this one over here to make those cuts here. And also from a previous video, I just also want to show you. I need to get them. Here it is. I think I'm not going to do that now, but I just wouldn't want to put those here under the microscope first, so, to show you. So what I've done is because this has to be clamped into the table, and as we 3D printed those specimen holders, just want to show you quickly how this actually works. And what I've done is the following. I've actually taken one of those here, put it in here. Yeah, they are basically have different diameters. Okay, like this here on the side, and then yeah, I put it in here. I think last week or 2 weeks ago I actually demonstrated this to you. Okay, and then you know what? I'm just gonna make 1 or 2 cuts. It's not stable, okay, because it's not clamped, right? I just for the sake of demonstration, and now you know why I've actually put it to the side, because it's a little bit of a support. And then, yeah, I better not cut myself now. And then basically what I've done is I've been cutting it, and then I've been advancing it 1, 2, 3, 4, 5, 6 times. And then I was able to, not enough, 1, 2, 3, 4, 5, 6. Yep, you can, yeah, have you seen? 1, 2, 3, 4, 5, 6. Okay, so you see that, yeah, it does kind of work. Considering the fact that the wood is actually, or was quite hard, I'm quite happy that it can be cut at all, right? So, and that's basically, and then I put it into water, and that's basically where we are right now, okay? So this is kind of, that's in water, yeah? And I just want to show this to you now as well. And we're just going to make a little, how do you call this, slide, again, as before. And I don't know, I'm just going to take this one here. So the bottom line is that heating it up and boiling the wood a little bit with some glycerin, apparently does the trick. I did a little bit of research. Oh, I'm always moving out of the picture. I did a little bit of research why glycerin should be used, is because it actually breaks the hydrogen bonds that actually stabilize the cell membrane, and therefore it makes it more softer. Yeah. But I found out as well that you have to boil it, or at least heat it up as well. Otherwise, it's not going to be quite as effective. So let's add a little bit of water here. And cover glass goes on top. Let's have a look at this under the microscope. So this is kind of a short tip that I found out. Is boil the wood in in glycerin to get it soft enough. Again, we have to wait until, here we go. I have to find the place here. Oh yeah. This one is not very nicely cut. Maybe the other one is, I don't know about this. Ah, yeah. Here it's a little bit thinner. Yeah. So we're able to see the individual cells here. Yeah. Yep. And here it's a little bit thicker here. Let's go down. It's a little too. Yeah, a non-stained everything, right? Yeah, so I think you get the idea, yeah, I think it looks nice. Yeah. So I'm just going to quickly have a look at a few comments here. I recently put a stem in alcohol. I didn't try it in alcohol yet. Okay. The oldest oak tree, that's what they claim. They say that it's the oldest oak tree in Europe, 1,000-year-old oak tree. Yeah, it's a nice looking tree for sure. But I mean, yeah. There's a little sign there, yeah, and it's apparently the oldest oak tree in Europe, and it's kind of yeah, a nice little place to visit, yeah. And I found some branches on the ground because, of course, I don't want to break anything off, and this is basically where I got those little pieces of wood from, yeah. Monocots have a solid center with dispersed vascular bundles. That's correct. However, this one here, oak trees are dicots. Okay, but maybe I'm not. Maybe this was a different conversation now. Okay. Okay, the discussion monocot versus dicot. I thought mono versus dicot was about the arrangement of the vascular bundles rather than the overall density. Yeah, that's yeah. Mostly dispersed. Okay, so that's a botanical issue. Okay. Check the message before the alcohol comment as it's relevant. Okay. Yeah. So this is basically just something where I was kind of wanted to show it to you as well, because I was actually able, after this glycerin treatment with some heating, I was actually able to make thinner cuts than before. Yeah, and it was easier to cut as well. Will make nice permanent slides. Yes. In any case, I still think that they're a little bit too thick, right? If you just look at this here, I mean, it does look nice, but it's still quite dark, right? So I'm not quite sure if we're able to see all of the details. I mean, after all, we're talking about wood here, which is, of course, has a very thick, strengthened cell walls. But one of the things that I kind of find interesting here is that even though it is not stained, you still see the different different colors. Yeah. So in that sense, yeah. So if I'm somehow able to get a very uniform cross section, maybe one without any cracks as well, like over here, yeah, then yeah, it would be a nice way to make permanent slides. Would this work for bamboo? Honestly, I have no idea. I never tried it with bamboo, but that's why we hobbyists like to try it out, right? So that's yeah. So here, for example, that's quite nice. Here it's a little bit thinner. But we can also see that there is still some cell contents visible as well. It's not just the cell wall, but also some cell contents. Yeah. But this one here was cut off at an angle, therefore it's getting thinner and thinner over here, until the whole thing is then kind of separated here. Yeah, so bamboo does soften when you steam it, so I don't see why it wouldn't work. Yeah. Does looking at this slide show us what makes oak hard? Well, maybe not by looking at this slide, but wood generally is hard because of a chemical called lignin. And it's actually the lignin that actually is the chemical that makes up wood. And if we actually look at kind of woody plants like this one over here, what we can see is that the cell walls are phenomenally thick. So those cell walls are quite thick, and they're not only thick, not only that gives a lot of strength, but there's also this lignin inside the cell wall. So while we're not able to see the lignin itself, it's simply the fact that we are able to see the thickness of the cells that already cell walls that already gives an indicator that it must be quite strong. Yeah. Thank you, Jack Kramer. Sent a comment. I sent an email with some tools to help with thin sections. Thank you very much. Also, thank you. I'll check. I guess sometimes I'm getting so many emails that I'm a little bit behind in reading all of them and answering all of them. Yeah, so please do be patient. Be patient. Yeah. Okay. Thanks from Altmünster. Oh, interesting. Yep, of course. He's yeah. It's nice to see that some colleagues, because I'm a teacher in school, some colleagues are watching, some students are watching, or former students are also watching this live stream. It's a nice way to meet to meet people again. Yeah, yeah. So essentially, that's something that I've been working on. I just want to show you something what you should not be doing. I don't know what I think is I don't know which type of wood that is. Oh gee, I forgot about this tube here. I'm gonna show you. This don't do that. Look, this is 50% glycerin, and I put some wood in here also to soften it. And what do you notice? Yeah, it got moldy. The thing is the following: if you actually have pure glycerin, yeah, then essentially organisms have a problem growing because the pure glycerin withdraws water. But if you mix it with water, then of course mold can grow. So this is something that basically I should not have done. Yeah, and you see it's pretty disgusting stuff here. So this means that yeah, you have to be a little bit. I should have. Been a little bit more careful, yeah, with that, because of course it becomes moldy if you, yeah, if it's organic substances are in here. Obviously, there's plenty of nutrients for fungi to grow. Yeah. So, okay, I'll put that back. But this was also a trial to kind of get the wood soft, yeah, and yeah. So the question is did you boil the wood in pure glycerin or diluted it with water? Apparently a 1-to-1 ratio of glycerin and water works well. Yes, this here is a 1-to-1 ratio of glycerin and water. But I did not boil it. I simply kept it in the tube, and that's why it got moldy. And therefore, what I've done then is I then put the wood into pure glycerin, simply to prevent it from turning moldy. And then it was still fairly hard. And then when I quickly heated it up in a microwave, it got very hot very quickly within a few seconds, but then it actually turned soft. And then I could cut it, right? So it wasn't, I wouldn't call it real boiling. I was just kind of heating it up for just a few seconds, and this was actually enough. So, yeah. So this is kind of a short comment here. And I think what I'm gonna do now is, yeah, it's already almost 1.5 hours. Yeah. And let's have a quick look again if the face mite is still alive. Yeah, that's the oak wood. Yeah, I have to. Yeah, it's of course always finding the specimen is always a yeah. That's the edge of the. Interesting. What are these round structures? Are these also some bubbles or something? Because they were not there before. Interesting. Let me quickly have a look. Oh, these are air bubbles. Why are the air bubbles there? Yep, here it is again. It's still the face mite still seems to be alive. I wonder how long I can keep it alive. I think I'm just gonna keep the slide. The advantage of oil is it does not evaporate, and for this reason, yeah, it should be active, stay active. Okay. Yep. I'm just reading again. Plant root hair are easy to section and show the difference between dicots and monocots. I have plenty of wood samples from the 50s, but nothing as beautiful as this oak one. Wondering why? Huh. Okay. I'm just reading. Okay. Yeah. No, I mean those wood samples that I've just shown you were basically Yeah, non-embedded. I think if you really want to have very nice cross sections, then it has to be properly embedded in in a matrix, and then using a proper under quotation marks microtome that is able to make very thin cross sections. I think then you're able to see the structures much better, right? Please answer my previous question. Do mites get into the butt? I don't understand this question. I mean, is the question if they have a butt? Because that's the thing is it's always been said is that these mites, face mites, they don't have an anus because somehow when they eat the food, the wastes are somehow deposited in the body itself. Okay, but do they get into the butt? Okay, you know what? I have a general, how do you say, philosophy. As a teacher, I try to answer every questions, and the answer is the following: it depends on the mite, and I'm quite sure that there might be certain parasites that indeed are able to enter certain body openings. As a matter of fact, there are certain worms. Parasitic worms, what they do is, I'm not making this up. Basically, they reproduce in the intestine, in the large intestine of the human. At night, they crawl out of the intestine. They can then be found in the underwear, and then they crawl back in. And that's why some children they always will scratch themselves on the butt because those parasitic worms they cause their butt to itch. Right? And some animals, some cats and some dogs also have this, right? That's why they're always kind of scratching themselves. And this is because of certain parasitic worms. And I'm saying, well, if parasitic worms are able to do that, maybe there are some mites that are also able to do that. Okay? So that's the answer of a biologist here. Okay? Yeah? So, I'm not making this up with the worms. That's indeed a case when, because children, sometimes they put everything into their mouth, also stones and gravel and sand when they play. And this is how they can actually also swallow some worm eggs, and then basically they will reproduce in the intestine, and they are able to crawl in and out. Yeah? So if it's possible with worms, then why not with other animals as well? Okay, yeah. So yeah. So this is something that I just wanted to comment on here, right? Honestly, when we talk already about mites, I will tell you before I say goodbye, the next research project, I'm now trying to find basically dust mites in my bed. I tried to sample already some dust from my bed sheets today, but unfortunately, I was not able to find any dust mites. Apparently, there are millions of them growing inside the mattress and inside the pillow, feeding on basically skin that we're always shedding off. Some people are allergic to the excrements of these dust mites, but unfortunately, my collection technique that I tried today was not able to. I was not able to find any. Okay, but what I'm going to do now is I think I'm just going to call it a day. Yeah. So technically, face mites and humans are a type of commensalism, right? It's not really mutualism or parasitism. Yeah. I would say that it's we humans. I guess we don't benefit and we don't have a disadvantage by those face mites, right? So in that sense, it's some evolutionary coexistence that we have, and yeah, I would say there are so many, how do you say, organisms growing on our body and in our body. Just if you look at microorganisms, bacteria, we have more bacteria growing on our body and in our body than we have body cells. So things like this, like those few face mites, they don't really surprise me at all. Okay, I think I'm going to call it a day for today. I think I'm going to, if everything works out, I'd like to again do a live stream like every Saturday, maybe next Saturday again. If not, then I'll of course make an announcement. And I'm going to call it, quits for today. And I wish you a nice week. Happy microbe hunting as always, and leave your comments, please. Right, and have a nice week. And bye bye.